Welcome back, everyone, to the deep dive. Today, we are going to get into something really cool. We're going to take a deep dive into a technique that's absolutely essential when you're working with biological materials. We're talking about solvent precipitation.
That's right. And the really interesting thing about this is it kind of sounds complex, but when you break it down, it's actually a very clever way of separating and purifying all sorts of things like cells and proteins. We were looking at some material that really highlighted how important this is in industrial biotechnology. They use it to get rid of all the unwanted stuff, the cells,
Huh?
and to really zero in on those valuable proteins that they're after.
Yeah, it's kind of like this hidden hero in the world of biotechnology. People don't always realize how much it underpins so much of what we do.
I totally agree. So our goal today is to give you a good grasp of how it all works, the core principles behind it, the practical steps involved, and the real-world applications. But of course, we'll also touch on some of the limitations because every technique has its pros and cons.
Absolutely, and that's what the deep dive is all about, right? Getting into the nitty-gritty. So let's start with the basics. Solvent precipitation. We're talking about using specific liquids to make these tiny biological components clump together and separate out from a mix.
Exactly. And the key here is understanding how these solvents actually influence the environment around proteins. So basically, when you add a water-miscible organic solvent, you actually disrupt that natural hydration layer that surrounds the proteins.
So it's like making them less comfortable in the water.
Precisely. They don't like it as much. And because of that, the parts of the protein that are hydrophobic, meaning they kind of shy away from water, they suddenly become more attracted to each other.
So it's kind of like if you drop some oil into water, the oil droplets all want to clump together.
That's a great analogy. And these strengthened hydrophobic interactions are what drive the whole process. They cause the proteins to stick together to aggregate. Eventually, they become heavy enough to precipitate out of that solution.
It's fascinating how such a simple concept can have such a profound impact on separating these molecules. Now, if we were to actually do this in the lab, how would we go about it? What does solvent precipitation look like in action?
So what you do is slowly add a cold organic solvent into the mixture. Usually something like ethanol or acetone that's been chilled way down to like -20 degrees C or even lower. And while you're adding this, you gently stir the mixture at the same low temperature.
I'm intrigued by that sub-zero temperature requirement. Is there a specific molecular reason why it's so important to keep things that cold?
It all comes down to protecting the protein structure. That low temperature minimizes the risk of denaturation, which is basically when the heat causes the protein to unfold and lose its natural shape and function. And adding the solvent slowly and stirring gently helps control the aggregation process and make sure it happens evenly.
So now you've got this mixture and you've got these clumps of stuff starting to form. How do you actually physically separate that precipitated material?
Well, there are a couple of ways to do that. One way is to simply let the aggregated material, whether it's cell mass or proteins, just settle to the bottom of the container. But another faster method is to use centrifugation. That's where you spin the mixture at really high speeds, and that forces the precipitate down to the bottom much more quickly.
Okay, so we're starting to get a really clear picture of the process itself. Now let's shift gears a bit and talk about why this technique is so widely used. What are some of the key applications where solvent precipitation really shines?
Well, the material we've looked at really emphasizes two major areas. First of all, it's really effective for removing the bulk of cell mass from a liquid. This is a crucial step in many bioprocesses. And secondly, it's a go-to method for protein precipitation, which lets you isolate and concentrate specific proteins from a complex mixture.
And it's not just limited to proteins, is it? I remember reading about ethanol precipitation being used for DNA and RNA.
You're absolutely right. It's actually a fundamental technique in molecular biology, and it just goes to show how versatile this principle is. You can apply it to all sorts of biomolecules, not just proteins.
So with all these different separation techniques out there, what makes solvent precipitation the preferred choice in certain cases? What are the advantages that make it stand out?
One of the big advantages is its effectiveness across a broad range of molecules. You can use it to precipitate a variety of proteins and other biomolecules. And as we discussed, that low temperature operation is crucial for preserving the integrity of sensitive biological materials.
Of course, no technique is perfect. What are some of the inherent limitations or challenges that come with solvent precipitation? What are the downsides?
That's a great question, and it brings up the issue of selectivity. One of the main limitations is that solvent precipitation is generally non-selective. That means it tends to precipitate out a range of molecules from the solution, not just your specific target. So you might end up with your protein of interest, but it'll be mixed in with other proteins and cellular components.
So it's like casting a wide net and catching a bunch of different fish instead of just the one you're after.
Exactly. And that means you'll likely need additional purification steps downstream to really isolate your target molecule.
So what other factors do scientists need to consider when using this technique?
Well, safety is a big one. Many of the organic solvents that are commonly used in this process are flammable. So that means you need specialized equipment that's explosion-proof to work with them safely. And even though we keep things cold, there's still a risk of denaturing some proteins, especially if they're particularly sensitive.
I imagine dealing with all those solvents after the process can be tricky too.
You're right, there's a whole other aspect to consider, which is solvent removal and recovery. It adds complexity and cost to the overall process. And of course, proper solvent disposal is essential for environmental reasons.
So taking all of that into account, where does solvent precipitation fit into the big picture of biological separations?
Well, when you look at it in context, the solvent precipitation is still a really valuable tool, especially for that initial bulk separation and concentration. But you're right, it definitely requires careful optimization and control to minimize any damage to your target molecules and to manage the risks associated with the solvents themselves.
Absolutely. So we've covered a lot of ground today. We've explored how solvent precipitation leverages these specific solvents to change protein solubility, leading to aggregation and separation. It's a really powerful technique, but it's clear that it requires a nuanced approach.
I think what's really cool is how a seemingly simple change in the solution's chemistry can have such a dramatic effect on how these complex biological molecules behave. It really highlights the intricate interplay between chemistry and biology.
It definitely does. And for you, our listener, we want to leave you with something to think about. Considering that solvent precipitation often brings down more than just your target molecule, what other purification techniques do you think might be essential for obtaining those truly pure biological products? That's something to ponder as we wrap up today's deep dive. We'll see you next time for another fascinating exploration into the world of science and technology.